Hey everybody, Mr. Grove here. Today we're going to talk about the processes of transcription and translation. So in the cells in your body, every cell except for your gametes contains every bit of DNA that you got. And so that means that somewhere along the line, a hair cell has to differentiate from being a red blood cell or an eye cell or a muscle cell. And they have to make special proteins unique to them. Even though all the hair DNA is in the eye cell and all the eye DNA is in the hair cell, we somehow have to specialize, and that is through the process of transcription and translation. So when we take our DNA of everything, we take one gene off of that DNA, and we can make RNA out of that, and then that RNA corresponds to one protein, and that can be very specialized then through that process. So let's take a look at RNA. There are three different kinds of RNA, mRNA, tRNA, and rRNA. That's messenger, transfer, and ribosomal. And some things that are different about RNA compared to DNA is, first of all, it's single-stranded. So DNA is double-stranded, RNA is just one strand. Second is it's way, way smaller. So DNA is all of the genetic material in your body, whereas RNA is just the genetic material for one gene. It would be kind of like if you have your Dish Network and you hit the guide and you see all the TV shows for all your channels for all week compared to just one show. So RNA would be like the one show. The entire TV guide would be like the DNA. The other thing is the name itself. So DNA is deoxyribonucleic acid, and that means this number 2 carbon does not have an oxygen there. But in the case of this nucleotide, this would be RNA, and our number 2 carbon does have an oxygen, and so that means it would be ribonucleic acid. So it's not been deoxygenized. And then one last little thing, it's actually a big thing, is we don't get to use T's. So there's no thiamine in RNA, and we use the nucleotide uracil instead. So once we have that, then it's time to do transcription. So transcription is basically the process of going from DNA to the RNA. It will take place in the nucleus. And so we've got to figure out where to start the transcription. And so I'm going to switch over to a little video here, and we'll kind of narrate as we go. And so the transcription is going to take place. So we have our promoting factors, and so we actually are looking for what's called the TATA box, TATA sequence, and these transcription factors will start then. And so once they hook onto the DNA, they will open up the double helix, and RNA polymerase will start to add RNA nucleotides on the template. So we will match up to the template, so that way the messenger RNA will have the same effect as the gene itself. And so that is going to be the process of transcription. So when we go back to here, when it's all said and done, we will have our messenger RNA. It will be the same as the actual gene because we matched up complementary to the template. We also have to do a little bit of stuff to get the RNA ready. There's a lot of junk DNA, what's called introns, and that's kind of like the commercials. So you recorded your show out of the giant network of TV guide, but you don't want to watch the commercials. And so we got to get rid of those introns. And so that's a way in which your body just shows its efficiency. Why would we make copies of nucleotides that really don't code for anything? And those introns can be in between genes. They can be in the middle of a gene. So once we get the introns moved, we're going to put a cap on and a tail, a poly A tail and a 5' cap. And this just kind of shores up the mRNA. It's about to leave the nucleus, and so it's going to be used. It's got to be strong and durable. And so once we got our cap and tail and got the introns out, that RNA, messenger RNA, is ready to go out into the cell. Where's it ready to go to? Well, that would be to the ribosomes. So we remember back in our organelle days that ribosomes are what produces proteins. And so our ribosomal units would be like the rRNA. And so the messenger RNA threads through that like a typewriter ribbon through a typewriter. And then we have these slots then that these little tRNA molecules can come in and fit into. And so the tRNA has an anticodon that complementary matches up with the mRNA's codon. And depending on what anticodon that is, tells you which amino acid you will carry with you. And so let's try and switch back over. So if we watch our tRNA grow, so here is our mRNA, and then we're going to have our ribosomal subunits come and form the ribosome. And once that happens, then we have to figure out where the initiating site would be. And that first tRNA comes in, and it has the anticodon that matches up to the codon on the mRNA. And so once that fit matches correctly, it brings the specific amino acid with it. And so that first one is usually the start amino acid, which is also the methionine. And the subunit will slide along the mRNA and allow another tRNA to enter into the next slot. So our next tRNA has an anticodon, matches up to the code on the mRNA, and brings a different amino acid with it. And those amino acids, while they're side by side, then will glue together through what's called a peptide bond. And this continues on, growing as we bring more tRNA molecules, more anticodons matching, more amino acids. And when it's all said and done, we end up with a long string of amino acids that then can be folded into a protein. And so that is the process of translation. So we can kind of summarize: we initiate it, we elongate the protein amino acid sequence, and then we terminate it when it reaches a terminating codon there. So let's take a look at a little practice then. If we have a gene to start with, so this would be our gene, we would want to make sure we could write our complementary DNA. So remembering our little acronym at Greenville College. So A's go with T's and G's go with C's. So in this case then, our ATG would turn into TAC. Opposite the C would be a G, opposite CG, opposite the G, a C. GGG would be CCC, TAT, ATA, TAG, ATC. So this would be our double helix. So the gene side and then the template side. Once we go into the first transcription to make our mRNA, so that would be opposite the DNA template. So opposite a T is an A, opposite an A is a T. However, RNA doesn't use T, so we have to put a U in there. Opposite a C is a G, opposite a G is a C, a G to a C, a C to a G, a C to a G, a C to a G, C to a G. And opposite an A would be a T, but again, we can't use T's, so we got to put a U. Opposite a T would be an A, and opposite an A would be a T, but we got to put a U again. And an ATC, the opposite of that would be UAG. So this is our mRNA. Note that it matches almost identical to the gene. ATG, AUG, CCG, CCG, GGG, GGG, TAT, UAU, TAG, UAG. And so that's what the mRNA does. It has the message of the gene. When we go opposite the mRNA for our tRNA anticodon, then that would give us this. So UAC, GGC, CCC, AUA, AUC. So that's opposite the mRNA, which would be the same then as the DNA template. So the template is the same as the tRNA. And you can kind of remember that T for template, T for tRNA. Then finally, when we go to write the code for our amino acids, on that one, we have to use the mRNA. It's a very common mistake for students to just get cruising along. They get to the tRNA, and then they jump over to their code table using it. But the message of the gene is in the mRNA. So once we've got that, then we can do our amino acid code table. So AUG, we find where that's at. So AUG would be our methionine or the start. And you can write either one is acceptable there. Then we have CCG. So we go to CCG and we get proline. Okay, so proline, CCG. Then we go to GGG, okay, which would be glycine, GGG. Then we get UAU, so UAU, which would be tyrosine. And then let's do that here. And then we have our last one, UAG. Well, that's one of our stop codons. So that tells us that, hey, we're done. We've made the amino acid sequence, and now we can start to fold. And you can just write stop on that. So when it's all said and done, filling out our table, these would be the sequence of the genetic code. Definitely you would not have to memorize the code table. That would always be provided for you. And just as a reminder, the two main mistakes, always use your mRNA for your amino acids, and for RNA we can't have T's. So double check, make sure, yep, no T's there, it's all U's, so we should be good. So I hope that was helpful, and smash that like button.